Welcome to another episode of the MEGIN Spotlight series. Today we are delighted to have as our guest Dr. Alex Wiesman, NIH Postdoctoral Fellow at Montreal Neurological Institute and Hospital. Hi Alex, thank you for joining us today.
Absolutely, thanks so much for the invite.
So we recently had an introductory call and it was fascinating to hear about your journey in MEG and how your research also showcased the variety of MEG utilization in research in Alzheimer's and Parkinson's disease. So let's dive straight into the first question from our end. Can you tell our audience how you first became exposed to MEG in your studies and what made you want to pursue this as a career path?
Yeah, absolutely. So in my undergraduate education, I started out working entirely in wet labs. So I worked first in a behavioral endocrinology lab, and then I moved into a retinal electrophysiology lab, again, both during my time in Omaha. Very, very detached from the world of MEG originally, but as I started my PhD program at the University of Nebraska Medical Center in Omaha, I rotated through Tony Wilson's lab, who does MEG research primarily, and I really just kind of fell in love with the big picture types of research questions that we were able to ask with MEG data. Knowing full well that I had absolutely no experience in computer science and signal processing and most of the things that are necessary to do MEG research, I saw this as a really exciting challenge, and I took the leap, and I'm really glad that I did.
How did you go from studying behavioral endocrinology to where you are now?
Yeah, so it definitely seems like a bit of an odd career path. On paper, I can at least justify it as being relatively straightforward. So I started again, collecting fecal samples from zebra finches, and I moved into studying human brain activity with MEG. I kind of started off again in a wet lab, so I was doing a lot of pipetting, PCRs, ELISA assays, things like that. I kind of leveraged my expertise in that area to get a job studying single cell retinal neurophysiology with Wally Thorson in Omaha during my undergraduate degree. And then from there, I can kind of make the connection that I applied what I learned from that single cell neurophysiology research to neurophysiology research on a much larger scale with MEG, right? It's very similar looking signals in some ways that we're looking at, but just recorded from many more sensors. In a very different environment, obviously. So I can kind of make that link from one to the other, but if I'm being honest, I think it was mostly serendipitous. I really enjoyed cognitive neuroscience topics when I was doing my undergraduate studies, but I think I had the misperception that I think a lot of undergraduate students do, that fMRI was kind of the cutting edge technology for this kind of research. And since there weren't really many options for fMRI research at that time in Omaha, I kind of, thought if I'm going to stay in neuroscience, I have to stay at the cell molecular level. I have to stay in the wet lab. But then once I started looking at PhD programs in the area, I became familiar with Tony's MEG work. I kind of, was opened up to this whole new world of cognitive neuroscience research with MEG. And once I rotated through his lab, I was completely hooked.
So some of the challenges people face in breaking into MEG or starting MEG programs is in learning how to do the data analysis. When we spoke to you, you told us that you collaborated with many mentors, some being Tony Wilson at Boys Town, for example, and the MEG Bootcamp and MEG Key Opinion Leader, Sylvain Baillet. Can you tell us a bit more about how that influenced you and your own experience and how you learned data analysis, since that is such a challenge in this industry?
Yeah, absolutely. I think you hit the nail on the head. One of the biggest barriers to entry for MEG, especially in a research setting, is just the complexity of the data analysis and sort of finding a way to learn those different techniques. I think this honestly stems from what's also one of the biggest benefits of MEG inherently, which is the extremely rich nature of the data that we're dealing with. So, we're sampling with sub-millisecond accuracy oftentimes across hundreds of sensors with the ability to then, if we want to, source image the data, which then increases the number of spatial features we're dealing with into the thousands or even tens of thousands. So very quickly, even within just a single individual, we're looking at millions of data points oftentimes. And again, that's one person, right? Typically, we're then doing studies of dozens or even hundreds of individuals. So, this gives us access to neurophysiological activity across the entire cortex at essentially any temporal scale that we can justify theoretically, alongside things like frequency, defined activity, or connectivity metrics, which again, just increase the number of features we're dealing with. So this leads to a situation where there really almost can't be one standard single approach to MEG analysis. That's a bit of a blessing and a curse, right? It's very exciting to be able to ask so many wide-ranging questions of our data, but at the same time, it makes it very difficult to sort of dip a toe into the waters of MEG analysis and learn how to do analyses like what you're seeing published in papers. Personally, I definitely had that kind of a shock experience when I first started in the field. I joined Tony's lab and I started reading papers that are published in Nature and really high impact journals, and I was daunted but very excited at the same time by the complexity of what was being done there. So from my own experience, I think the best advice I can give is to start with the basics, learn from the ground up. You need to be able to pre-process data and make sure the data is clean and of high quality before you move on to fancy connectivity analyses or cross-frequency coupling analyses. So yeah, start basic, learn to process data, perform basic sensor level analysis, perform basic dipole analysis, things like that, and then kind of branch out from the basics. I think it's also really helpful to learn standard analyses when you have them on a single participant first. So oftentimes, again, we're going to be looking at data sets of tens, if not hundreds of individuals. So you want to make sure your data analysis pipeline runs effectively on a single person before you try to implement it on a large group of individuals with a batch. So starting small and then working your way up is kind of the general advice I'd like to give. But then also, I think probably most importantly, it's important to look for sources of expert support. So, most commonly this will be local. So these will be mentors or advisors that are able to sort of give customized advice based on your research questions. Again, a lot of the time what we're doing with MEG is asking questions that haven't been asked before, which requires analyses that, at least in some ways, diverge from what's been done previously. Because of that, I think it's really essential that you have someone that's been in the field for a while, that really understands the nuances and the limitations of the method. And that way they can give really detailed feedback. Again, it can be local, it can also be virtually now, especially, or long distance. But getting that expert feedback can be really essential. And then from there, there's also something that to be said for the support that's available through all of the major open softwares. So softwares like Brainstorm, Fieldtrip, M&E, and many more, they have really great tutorials. They supply tutorial data sets that you can work on, and they have really good support forums. So oftentimes, if you're looking at this tutorial data set and you're running their sort of basic analyses that they recommend, you can actually benchmark what you're doing to what's been done previously on the same data. And that gives you a really good reference point before you venture out into the unknown with your own data.
So you were in good hands then with Tony and Sylvain, I would think.
Absolutely, yeah, trained by two of the best.
Yeah. So can you also tell us a little bit about any sort of specific recent or upcoming studies you've been involved in, and also where MEG was utilized and how data was retrieved from MEG technology, which has helped you in the study and the findings?
Yeah, absolutely. So broadly, most of my recent work has used MEG to study patients with neurocognitive disorders. So these will be neurodegenerative disorders like Alzheimer's disease, as well as things like Parkinson's disease, HIV-associated neurocognitive deficits, things like that. And I think one of the clear benefits of MEG broadly in these kinds of studies is how amenable the patient groups are to the recording procedures and the environment of MEG scanning. A happier participant always provides better data, is also more likely to return for longitudinal studies, and it's just nice to keep people happy. So generally, I think MEG is really good for those kinds of studies, and I've seen those benefits myself. I think there's also obviously very clear benefits regarding the specific signal of MEG and what we're able to record in terms of data. So as most people watching this probably know, with MEG, we can study not only the time-locked sort of increases or decreases in neural activity generally to an incoming stimulus or things like that, But we can also study frequency-defined patterns of brain activity. And these frequency-defined or rhythmic patterns of neural activity are known now to differentially relate to specific cognitive and sensory motor functions in the brain. So in my work, we've used this to look at incorporating information not only in single frequency bands, but also across multiple frequency bands at once, which we found can actually provide additional information that isolated frequency band analyses cannot. So a great example of this is that in both patients with Alzheimer's disease and Parkinson's disease, we've seen that there's this multi-frequency shift in neurophysiological activity from faster activity to slower activity relative to healthy adults. So what we're seeing is in the same patch of cortex and the same set of cells, we're seeing this slowing effect where brain activity is becoming slower in these regions. And this effect seems to predict important clinical features in both of these disorders, things like cognitive impairments in Alzheimer's disease, motor impairments in Parkinson's disease. But it also seems to predict things like accumulation of harmful proteins in Alzheimer's disease across the brain. So the unique combination of high spatial and temporal resolution of MEG data is really makes it the only neuroimaging technique that we could have used to be able to make these findings. So very excited to be able to work in the area.
Thank you. How is MEG then used in conjunction with other neuroimaging modalities offered at the Montreal Neurological Institute? What type of research requires sort of multiple modalities as well?
Absolutely, yeah. Like any method, MEG has some limitations. So we oftentimes can combine MEG with other neuroimaging modalities to sort of leverage the unique capabilities of each and enhance the overall findings. Probably the easiest example that I can come up with is oftentimes with MEG, we want to image the cortical activity that we're measuring to the level of the cortex, to the level of the brain, using algorithms like beamformers or minimum norm estimation. And to do this as accurately as possible, it's preferable to collect a structural MRI alongside the MEG data so you can actually enhance the spatial specificity of your analyses. So that's a very common one where you can see different modalities combining to enhance the overall results. But at the MNI, we're also actively working on a number of other projects that are a bit more advanced in the multimodal combination of things. So one good example is a study where we're combining MEG with PET scans, so positron emission tomography, where we're combining those to look at how the accumulation of harmful proteins in the brain of participants at risk of Alzheimer's disease seems to affect neurophysiological activity. So we're looking at things like amyloid beta, tau, which are two of the most common proteins that are implicated in Alzheimer's disease pathology. And we're seeing how the accumulation of those proteins affects neurophysiological activity in the brains, again, of these participants who don't have Alzheimer's disease. They're just at higher risk for the disease, with hopes of being able to then sort of track neurophysiological changes preclinically in this kind of a disorder. Another good example is a study we're conducting using specialized MRI sequences to look at degeneration of deep brain structures in patients with Parkinson's disease. Oftentimes, these are the brain structures that are most commonly implicated in the disorder itself, the sort of death of neurons or brain cells in these structures. But it's really not certain in the field how the degeneration of these deep brain structures actually impacts activity at the level of the cortex, neural activity at the level of the cortex, and then how that leads to changes in cognition. So that's what we're working on in Parkinson's disease as well here. And again, I think that these types of multimodal imaging studies are really essential. They allow us to see direct interplays between neural activity measured by MEG and things like cytoarchitecture, molecular changes, and hemodynamic changes in the brain, just to paint a much fuller picture of brain function and dysfunction in pathology.
In your work with study participants with dementia and Parkinson's and HIV-related neurocognitive disorders, how do these patient groups respond to the experience of an MEG scan? You said you liked happy participants.
Yeah, happy participants are the best participants. I think it's like I alluded to before, all other things being equal, if we're talking about the scanning environment, MEG is absolutely my technique of choice for patient populations who have cognitive impairments. And again, this is largely due to the procedures in the recording environment. So Good comparisons would be compared to EEG. MEG requires very little pre-scan setup, which can oftentimes be a bit anxiety inducing for patients with early stage dementia or even just mild cognitive problems, while still providing better spatial resolution than EEG. And unlike MRI, MEG is quiet, less claustrophobic, and oftentimes even allows patients to stand up, to go to the bathroom, to speak with a confidant that accompanied them to the visit in between scannings. So altogether, this makes for an environment where patients are much less stressed, much less cognitively fatigued, which is oftentimes a bit of an issue for patients with early stage dementia, especially, right? So oftentimes they're a bit anxious and disoriented coming into a study for the first time. They're in a new environment performing tasks they've never seen before. So it's really essential that we don't exacerbate this and that we make the whole experience as low stress as possible. So, this is partially altruistic. It leads to a more comfortable experience for the patient. It also means that they can tolerate lengthier recordings. They can perform more demanding tasks when they're not as cognitively fatigued. And overall, it just contributes to a study environment where we can collect more interesting data, more detailed data, while also keeping the visit a positive experience for the participants, which helps with retention for, like I said before, longitudinal studies, but also just a nice thing to do.
And are there any areas of your current research you feel may support new clinical applications for MEG in the future, which is something that obviously everyone is sort of anticipating in this industry as well?
Yeah, I sure hope so. In the near future, I think some of our more recent findings that I've already discussed a bit might be applied to diagnostics and to measuring therapeutic outcomes in patients with neurodegenerative disorders. Basically, we're hoping that MEG might represent a lower cost and less invasive alternative to tracking disease status in patients with Alzheimer's, with Parkinson's disease. But we're also finding that MEG, like I said before, when collected early on in the asymptomatic or preclinical stages of Alzheimer's pathology, may actually be useful in predicting eventual susceptibility to disease factors and subsequent cognitive declines. So we think that has some very exciting implications for prognostics as well. So determining early on whether someone is potentially going to progress to cognitive problems, as well as potentially even when. But then beyond potential applications as a biomarker, I think we're also hoping that some of the neural changes that we see in patient groups with MEG might be modifiable using things like non-invasive brain stimulation, like transcranial electrical stimulation or transcranial magnetic stimulation, or even with things like rhythmic sensory entrainment. So there's been some really interesting animal research that's come out recently that shows that sort of rhythmic sensory stimuli, things like just a flickering visual stimulus or a rhythmic auditory tone, can actually help clear Alzheimer's disease type pathology in the brain. So we're hoping that maybe we can actually use some of what we're finding now to better target some of these types of stimulations to the specific brain regions and the specific brain frequencies that might be affected in these diseases. That said, this work is all somewhat nascent, so stay tuned. It's a key interest.
We'll definitely be watching. So then we're coming to our last question for today is why is working with MEG a rewarding career path? So what advice would you offer other researchers looking to enter the MEG field?
Yeah, so even though I kind of stumbled my way into the field, like I said before, I'm very happy that I did, and I wouldn't have it any other way. MEG allows scientists a level of analytical flexibility, I think, that's really engaging and challenging in the best way possible. So I like a challenge. I like struggling a bit with my research every once in a while. So I can't imagine really being as professionally fulfilled in other subfields as I am here. As far as advice, I'd say find a research group whose work really interests you and reach out to them if you're an early stage trainee. The MEG community has generally been very collaborative and very supportive in my experience. And there's a lot of MEGers out there from what I've seen who love to just talk about new ideas for study design, task design, and analysis approaches. So reaching out to people is probably the best advice I can give. And also don't be intimidated if you're new to the field by not having previous experience. At least historically, MEG hasn't been as nearly as common as something like fMRI or EEG in the research setting. So most research groups I've interacted with are very much used to training people from the ground up in study design and from data collection and analysis. All that's really required of you is motivation and scientific passion to get started. And I'm, I think, a good testament to this. I joined my first MEG lab with pretty much nothing but great pipetting skills. Yeah, absolutely, it's a great field to work in.
Thank you very much for speaking with us today, Alex. We really enjoyed hearing about your research work and utilizing MEG, and yeah, just thanks again for your time today. I appreciate it.
Absolutely. Thanks so much for having me.